Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, good evening. Today we shall discuss about two different enzymes. These enzymes are S1 nuclease and polynucleotide kinase. First of all, we shall discuss about S1 nuclease. S1 nuclease is an endonuclease that splits single-stranded DNA and single-stranded RNA into 5-phospho-oligo or mononucleotides. S1 nuclease is a glycoprotein. It is monomeric. Its molecular weight is 38 kilodalton. This enzyme requires 3 zinc ion H cofactor. It has a common active site. It requires a acidic pH. It functions optimally at pH from 4 to 4.5. It contains 3 glycons that are attached to asparagine residues via N-glycosidic bond. Now we shall discuss about the function and application of S1 nuclease. For S1 nuclease, the primary substrate is single-stranded RNA or single-stranded DNA. That means mainly S1 nuclease degrade single-stranded RNA and single-stranded DNA to release nucleotides. In addition to this, S1 nuclease also introduce single-stranded break in double-stranded DNA or double-stranded RNA or DNA-RNA hybrid. This is a double-stranded DNA, and in this double-stranded DNA, this single-stranded break has been created by the enzyme S1 nuclease, but occasionally. Next function is S1 nuclease hydrolyze single-stranded region in duplex DNA, for example, gap and loops. In this structure, this is the loop. Such type of single-stranded region are also cleaved by the enzyme S1 nuclease. For S1 nuclease, there is no need of sequence specificity. It do not require any specific sequence to act. This enzyme also cleaves a strand opposite a nick on the complementary strand. In this diagram, in this strand there is a nick. This is the nick and this is the complementary strand. So in such a double-stranded DNA in which there is a nick in one strand, in such a DNA, S1 nuclease can cleave the complementary DNA opposite to the nick. That means at this point this enzyme can cleave. So two DNA molecules will be formed. The enzyme S1 nuclease is present in the fungus Aspergillus oryzae. Then what are the applications of this enzyme? S1 nuclease is used in nuclease protection assay. This enzyme can also be used to remove single-stranded tails from DNA molecules. For example, this is a DNA molecule and these are the single-stranded tail. Such type of single-stranded tail can also be removed by the enzyme S1 nuclease. And another function is during the synthesis of DNA from mRNA with the help of the enzyme reverse transcriptase, a hairpin loop is formed. Such type of hairpin loop which is generated during the reverse transcription can also be cleaved by the enzyme S1 nuclease. This is about the application of the enzyme S1 nuclease. Another enzyme is polynucleotide kinase. This enzyme catalyzes the transfer of gamma phosphate from ATP to the hydroxyl group that is present at the 5' end of DNA or RNA. This enzyme is usually obtained from T4 bacteriophage or T7 bacteriophage. This is the reaction that is catalyzed by the enzyme polynucleotide kinase. This is a double-stranded DNA molecule, and you see these are the two 5' ends. And at this two 5' end, there is hydroxyl group. To such type of 5' end, phosphate group can be added with the help of enzyme T4 polynucleotide kinase. So in this double-stranded DNA, at the 5' end, phosphate groups are present, and these phosphate groups are added with the help of the enzyme T4 polynucleotide kinase. Polynucleotide kinase also has several applications. First application is polynucleotide kinase is useful for preparing DNA fragments for ligation if they lack 5-phosphate group. Other application is this enzyme is also useful for radio labeling DNA. For radio labeling DNA, we have to use the ATP in which the phosphorus of the gamma phosphate is phosphorus 32, and this process is useful for preparation of hybridization probe. Friends, this is about the enzyme polynucleotide kinase. Please like and share my video, and also subscribe my YouTube channel Sam Online Class, and also give your comments. Your comments will be helpful to me to improve the quality of my videos. See you friends, bye bye.